to now we've been discussing the passive processes that move materials from one side of a membrane to another. In this video, we're going to be discussing active processes that get stuff to move from one side of the membrane to the other. So you've seen this slide already. These are descriptions of the passive processes. I want to remind you again that active processes expend ATP energy. They're incorporated, or they are performed by living organisms, so these are living processes, and in these processes there will be changes to the cell and to the proteins associated with the cell. And once again, the cat is back. Let's see, get him into his resting spot. There we are. Okay, so they include active transport, endocytosis, exocytosis, and then a process called transcytosis. So active processes are required for materials that are either too large to pass through the membrane, can't dissolve in that lipid bilayer, or must be moved rather than down a concentration gradient, that is, from a high concentration to a low concentration, again, will be moved instead up the concentration gradient, or against that concentration gradient. So ATP is required because all of these things are working against the physical properties that would move materials if the conditions were appropriate. So how do we do all this? The first process that we're going to look at is called active transport. And active transport is very similar to facilitated diffusion in a couple of ways, except, and the main difference here, is that we're moving materials rather than from high concentration to low, we're moving against the concentration gradient. That is, from a region of low concentration, so if you look here at the drawing, to in the direction toward a region of high concentration. It requires a transport protein, very similar to the transport protein setup that we discussed when we talked about facilitated diffusion, and it requires some ATP energy. So ion pumps are an example of active transport, and they move charged particles like sodium, potassium, calcium, chloride ions. These are all moved when they are needed to be moved by these things called ion pumps. And because the ions have a negative or a positive charge, they cannot pass through that lipid, that nonpolar lipid bilayer. So what happens is that on the low concentration side, if this is say a sodium pump, sodium would find its way into, it would diffuse and find its way into this carrier protein. It would bind there, so that's a site that is attractive to the sodium ion. It would bind there. And then if we give some ATP energy to this carrier protein, it will change the shape of the protein in a way that will release the sodium ion here on the high concentration side. And so with ATP energy, we can get that energetic change in the protein that will move that material to the far side of the membrane. So, like I say, there are a couple of different kinds of sodium pumps. Ion pumps. There's just sodium pumps. There's another protein called the sodium-potassium pump, and it does an exchange so that it moves. I don't know why I have Cl minus. That should be a K plus. So it exchanges the sodium that's found inside a cell with potassium that's found outside a cell. So for every ATP you give it, you can move 3 sodiums out and 2 potassiums into a cell. And then there's the calcium pump that brings calcium ions into a cell. So these are all different kinds of pumps, ion pumps, that perform active transport. Another active process that moves materials into a cell would be, and across a membrane, is what's called endocytosis. So this brings in molecules or particles, even food bits, that are very large and cannot enter by diffusion and don't have a carrier protein that will bring them into the cell. What happens then, because the plasma membrane has this liquidy character to it, it can, when a material comes up near to the cell, the plasma membrane can be folded inward, so the cytoskeleton changes somewhat, and we see a fold inward that draws that material into the fold deepens and becomes a pocket, and eventually the pocket pinches off, and now inside of the cell we have a little vesicle, so it's a bubble of plasma membrane that has this piece of material that's been brought into the cell. Here we talk about it as being phagocytized. When you phagocytize something, or when a cell does that, it is eating. Phagophagy means eat. So this is how a cell can bring in a large particle in order to destroy it or to break it down, digest it into nutrient particles that can be used by the cell. So endocytosis brings materials into the cell if they are solid material, and endocytosis there are two ways that it can happen. If what we're bringing into the cell, rather than being a solid thing, is just a bubble of fluid, we refer to that as Pinocytosis, sometimes called cell drinking. And then phagocytosis brings in the solid materials. And generally, so most cells do this cell drinking, this Pinocytosis, and they just absorb the water into the cytoplasm, and then this bubble, the empty vesicle, can be bumped up here against the cell membrane. It can fuse with it and just return to the cell membrane. With phagocytosis, when solid materials are brought in, it has to be digested, and so that's when a lysosome can be very helpful. The lysosome will fuse with this vesicle that we make, and all of the enzymes will attack the food particle, break it down into small nutrients that can then be absorbed into the cytoplasm. So that's what we're seeing here in this third drawing associated with phagocytosis. And we have some very active phagocytes in the body. The white blood cells do a lot of phagocytosis, and they will cruise throughout the body looking for things that don't belong, consuming them and destroying them. There's a second type of endocytosis that, rather than just getting anything that's found outside of the cell membrane, it will actually bring in very specific materials. And it's referred to as receptor-mediated endocytosis. Receptor-mediated because we can place, or the cell can place receptors, these are specialized proteins, that have a spot on them that is very attractive to a specific molecule. And if that's a molecule that the cell wants inside of it, then it can put these receptors out there. The receptors will selectively attract and bind the molecule that they want, and that molecule, when that binds, are referred to as is a ligand, something that will be attached. So once all the receptors in that area of the cell membrane are full, so they've now have this receptor ligand complex or combination. Once all the receptors are full of ligand, then and again, the target molecule that's a general term for something that will bind to a receptor, is ligand. It doesn't mean a specific material. So that ligand then is that stretch of the cell membrane that has the receptor ligand complex attached to the membrane will do endocytosis. So now we bring in a little vesicle that contains exactly what we want and leaves outside of the cell the stuff that we don't care about having inside the cell. So this receptor-mediated endocytosis allows for us to, or for the cell, to very specifically target the molecules that it would like to bring into the cell. And then, this is kind of cool, we can just, again, bump it up to a lysosome. The lysosome will break away the ligand from the receptors, digest the ligands only. The receptors remain in this vesicle, and we can bring that vesicle back to the cell surface and boop, just do this process called exocytosis. I know we haven't talked about it yet, but it lets us then bring those materials into the cell. I'm sorry, I lost what I was saying. By sending the empty receptors then back to the cell surface, we can stick out more receptors to be able to collect more of the ligand that we're interested in binding and bringing into the cell. Okay, so this is an example of material that's brought into the cell this way. Includes insulin. We bring in cholesterol this way, and there are some cells that will bring iron into the cell this way. So this receptor-mediated endocytosis is a relatively common way of bringing in very specific materials to the cell. Okay, so we can get stuff back out as well, and that process, if bringing it in, is called endocytosis. Ejecting it from the cell is called exocytosis. Exit, X means outside, so exocytosis takes materials that are products of the cell and pushes them outside the cell. So, for instance, some of the stuff that we might push out would be mucuses that the cell produces, hormones that the cell may be making, neurotransmitters that are being released by a neuron to communicate across to other cells, because that's how neurons communicate, is by releasing these neurotransmitters. We may dump out wastes from phagocytized materials from the cell, so it's sort of like makes a little trash can and pushes the waste out. But here's the way that we do it, and here we're going to follow a pathway that is all about a product that the cell has made being released. So if the endoplasmic reticulum produces a material, then that will be delivered to the Golgi apparatus. That material will be modified in the first pancake of the Golgi apparatus, delivered to the second pancake, and so on until we have a final product produced, and that final product will be wrapped up in a vesicle, which we see here at the tip of the little arrow that I drew. That vesicle can be bumped up against the cell membrane, and again, because of the fluid nature of the cell membrane, the vesicle membrane and the cell membrane will fuse together, and that will then place all the contents of the vesicle now on the outside of the cell. So but that's the cool thing about these cell membranes, is that they're fluid, and if you push one cell membrane against another, those phospholipids will interact with each other and assort themselves into a membranous unit, a single cohesive membrane, and in that way we can push things outside of the cell. So that's called exocytosis. And then there's one last movement of materials across cell membranes that requires ATP energy. And I forgot to mention that before, but endocytosis, receptor-mediated endocytosis, exocytosis, and transcytosis all require ATP energy in order to do this. All that rearrangement of the cytoskeleton and all of the movements of vesicles require ATP energy to accomplish that. Okay, so with transcytosis, this is a means of pushing a material directly across a single or a very thin layer of cells and moving it from the lumen, or from the opening of, say, the digestive tract or the lining of the anus or the vaginal canal, we can move materials directly across that lining and into the body. And it's done as a cell there at that lining surface does endocytosis, creates a vesicle with some material inside it, and rather than digesting that vesicle, and the contents of that vesicle, instead it just transports the vesicle across the cell and then bumps it up against the cell membrane on the other side, and then releases that material into the watery environment inside the body, into that membrane, or not membrane, but tissue fluid that surrounds all the cells in the body. So it's endocytosis, no digestion, transport across the cell, and then immediately exocytosis. And so this is how we absorb digested food in many locations of the intestine, and a number of different digested food materials are brought into the body this way. And it also occurs at the lining of the anal canal and in the vagina as the HIV virus is brought into body fluids. So it's through transcytosis that HIV virus particles are able to make it into the body. And it's a nice, convenient means for that virus to get inside. Now, hopefully, white blood cells will eat and destroy the virus, but that is also the scary piece about HIV, is that it actually targets the white blood cells that are responsible for doing that phagocytosis of those things. So, good information in the book about all of these processes. If you're still confused, I encourage you to try reading. If you haven't done that yet, listen to the video again, or you can send me an email, find me an office hour, or give me a call. So good luck with learning this stuff. Only one more video to go.